Good morning, our participants. I'm Surang Leelawat from Medicinal Cannabis Research Institute, College of Pharmacy, Rangsit University. Today, I would like to talk about the effects of medical cannabis. Cannabis preparations have been used medically and recreationally for many centuries. Nowadays, an increasing of scientific and public interest in the medical application of cannabis are observed. My presentation is focused on cannabis anti-cancer activity, which entitled Cannabis Extract Work for Cancer. Cannabinoids are chemical compounds from cannabis. They demonstrated a therapeutic potential in many medical fields. Nowadays, cannabinoids have found an application in the palliative medicine due to the analgesic and anti-emetic activities. In addition, several preclinical studies of cannabinoids show their anti-cancer properties. Today, I will focus on the current understanding of molecular mechanism of cannabinoid anti-tumor effects and our studies about the effects of THC on cholangiocarcinoma and the effects of CBN on lung human cancer cells. Cannabinoids are molecules that moderate the endocannabinoid system. Cannabinoids generally have been classified following their origin into endocannabinoids such as arachidonoyl ethanolamide or AEA and 2-arachidonoyl glycerol or 2-AG. Phytocannabinoids such as delta-9 tetrahydrocannabinol or THC, cannabidiol or CBD, and cannabinol or CBN. Synthetic cannabinoids such as THC, methamphetamine, and nabilone. For phytocannabinoids, THC and CBD are the most intensive study of cannabinoids in cannabis. THC is the principal psychoactive component and displays a number of biological activities including analgesic, anti-emetic, and cytotoxic activities. CBD is a major non-psychotropic constituent, shows analgesic, anti-inflammatory, anxiolytic, and anticonvulsant activity. For minor phytocannabinoids such as CBG, CBN, and CBC. The biological properties of cannabinoids are principally mediated by two members of the G-protein couple receptor family, cannabinoid receptor 1 or CB1 and cannabinoid receptor 2 or CB2. The majority of CB1 expressed in humans is originated in the brain, where it is complicated in various neurological activities. CB1 receptor also found on the peripheral sites in a lesser extent, such as liver, reproductive system, and skeletal muscle. CB2 is predominantly expressed in cells of immune system and is commonly expressed in peripheral tissue including liver, colon, pancreas. A well-known function of endocannabinoid system is its role in neuromodulation. After binding of neurotransmitters to their receptors, postsynaptic neurons synthesize and release endocannabinoid. The endocannabinoids bind to presynaptic CB1 receptor to block neurotransmitter release. This allows the modification of memory, movement, appetite, and pain. THC function in the body by activating CB1 receptor, the same as endogenous ligands. Cancer is the second leading cause of death globally and is responsible for an estimated 9.6 million deaths in 2018. Cancer is a group of disease involving uncontrolled growth of abnormal cells with the potential to invade, called local invasion, or spread to other parts of the body, called metastasis. The mechanism of cancer cell proliferation and invasion. Abnormal cell growth are uncontrollable. Cancer cells can move through the basement membrane to nearby organs and spread to distant organs, as well as induce angiogenesis. Finally, a mass of cancer cells develop. This figure shows the hallmark of cancer. It is the capabilities that enable tumor growth and metastatic dissemination. For example, sustaining proliferative signaling, evading growth suppressor, and avoiding immune destruction. The hallmark of cancer provide a basis for understanding the biological of cancer. All function capabilities are thought to be acquired by developing cancers during the multi-step carcinogenesis that leads to the form of human cancer. The order and significance of capabilities of their contributions to malignant disease appear to vary through the spectrum of human cancers. The description of hallmark principles is to develop therapeutic improvement. Drugs that affect each of the hallmark capabilities and hallmark enabling processes have been developed in preclinical and/or clinical testing. A focus on antagonizing specific hallmark capabilities is expected to develop the novel, highly effective therapeutic strategies. General mechanism of cannabinoid anti-tumor action. Cannabinoids impair tumor progression at different levels. Their most prevalent effect is the induction of cancer cell death by apoptosis and the inhibition of cancer cell proliferation. In addition, in vivo experiments have shown that cannabinoids impair tumor angiogenesis and block invasion and metastasis. General mechanism of cannabinoid anti-tumor action involved in three mechanisms. The first one is inhibition of uncontrolled cancer growth by induction of apoptosis. The second mechanism is sustaining angiogenesis by downregulation of VEGF pathway. And the final mechanism is inhibition of metastatic capability by downregulation of MMP2 and modulation of other targets. From hallmark of cancer, general mechanism of cannabinoid anti-tumor action is likely to involve in four capabilities: sustained proliferative signaling, resisting cell death, induced angiogenesis, and activating invasion and metastasis. The first mechanism of cannabinoid anti-tumor action is induction of cancer cell death. THC-induced cancer cells apoptosis by stimulation of the de novo synthesis of pro-apoptotic ceramide through CB1 and CB2 receptor. CBD is a phytocannabinoid with a low affinity for cannabinoid receptors. CBD enhances the production of reactive oxygen species in cancer cells, leads to autophagy-mediated apoptotic cell death, This figure shows molecular mechanism of apoptosis induction by cannabinoids. THC stimulate de novo synthesis of ceramide through CB1 and CB2 receptors. Accumulation of ceramide leads to an activation of stress signaling pathway. This involves enhanced expression of pH protein as well as its downstream targets. stress can lead to an activation of intrinsic apoptosis pathway. Activation of pH-regulated pathway cause inhibition of AKT by TRK. That in turn leads to an inhibition of mTORC1. And finally to apoptotic cell death via autophagy. In some cancer, inhibition of AKT leads to an activation of P27 and P21 protein, cause phosphorylation of retinoblastoma protein, and ultimately cell cycle arrest and apoptosis. In glioma, downregulation of AKT signaling leads to the reduction of bad phosphorylation, resulting in apoptosis. For inhibition of angiogenesis, cannabinoids block the activation of vascular endothelial growth factor pathway or VEGF pathway. VEGF and its main receptors, VEGF-R1 and VEGF-R2, are downregulated on cannabinoid treatment. For inhibition of invasion and metastasis, cannabinoids reduce the formation of latent tumor masses in animal models depend on the modulation of extracellular proteases such as matrix metalloproteinase 2 or MMP2. For anti-cancer effects of cannabinoids in clinical trials, the first experiment was a pilot clinical study on 9 terminal patients with recurrent glioblastoma who were resistant to the standard therapy. Patients received THC intratumorally. The study indicated a good safety profile for THC and without overt psychoactive effects. Median survival from the beginning of cannabinoid administration was 24 weeks. And then phase 2 placebo-controlled clinical study of a combination of THC and CBD in 21 patients with recurrent glioblastoma were performed. The findings from the study suggested that adding of a combination of THC and CBD to patients on temozolomide produced significantly improvements in survival compared to placebo. The cannabinoid treatment was generally well tolerated. My first studies is the effects of THC on cholangiocarcinoma. This publication shows that THC inhibits cholangiocarcinoma proliferation and migration. Cholangiocarcinoma is a malignancy of biliary duct system. It originates in the liver and extrahepatic bile ducts. Cholangiocarcinoma is one of the most common cancers in the population of northeast Thailand and responsible for 1 in 5 cancer-related deaths among Thai patients. In Thailand, liver fluke infestation is the main cause of cholangiocarcinoma. Most people have advanced stage disease at the time of diagnosis. Currently, only gemcitabine plus platinum demonstrates the considerable activity for cholangiocarcinoma. Cell proliferation assays were performed in cholangiocarcinoma cells treated with THC at concentration of 5 to 100 micromolar or vehicle. After 24 hours of incubation, the results show that THC at 5 to 20 micromolar has no significant effects on the inhibition of cholangiocarcinoma cells proliferation when compared to untreated cells. However, at high concentration of THC from 40 to 100 micromolar, cholangiocarcinoma cells were significantly inhibited in a dose-dependent manner. A tunnel assay were performed to confirm the mechanism of THC-induced cholangiocarcinoma cells apoptosis. THC or vehicle were added to cholangiocarcinoma cells and the cells were incubated for 24 hours. This figure shows cholangiocarcinoma cells under fluorescence microscope. Cholangiocarcinoma cells pre-treated with 40 and 100 micromolar THC show apoptotic cells in green spot. The results show that the number of apoptotic cells after exposure to 40 and 100 micromolar THC was significantly increased. And then cholangiocarcinoma cells were treated with THC or vehicle for 24 hours and migration and invasion assays were performed. This figure shows invasive cells that can pass through matrix cells and were stained by hematoxylin. At 20 micromolar THC, cholangiocarcinoma cells migration and invasion significantly decreased compared to untreated cells. The morphology of cholangiocarcinoma invasion. Inactive state cholangiocarcinoma cells show epithelium-like cells with characterization of cell-to-cell contact and no polarity. However, active states of cholangiocarcinoma cells show mesenchymal type with loss of cell-to-cell contact and has polarity. The change of morphology of cholangiocarcinoma from epithelium-like cell to mesenchymal cell called epithelial mesenchymal transition or EMT. This figure shows actin polymerization under fluorescence microscope. Mesenchymal cells show high level of actin polymerization and pseudopodia formation. We next investigated actin polymerization, which is implicated in cell mortality mechanisms. Cholangiocarcinoma cells were stained with phalloidin to detect actin polymerization. The upper figures show cholangiocarcinoma cells under fluorescence microscope. Cholangiocarcinoma cells treated with 20 and 40 micromolar THC show lower extent of actin polymerization. The lower figures show cholangiocarcinoma cells under confocal laser scanning microscope. Cholangiocarcinoma treated with 20 micromolar THC show a loss of stress fiber and philopodia compared to untreated cells. Anoikis is a form of cell death. Anoikis resistance involved in cancer metastasis. The inhibition of anoikis resistance to cholangiocarcinoma cells, which could reduce their ability to metastasize. In order to investigate the ability of THC to inhibit anarchist resistance in cholangiocarcinoma, cholangiocarcinoma cells were seeded on polyhematocytes and treated with THC. Cell viability were determined at 6 and 12 hours after seeding. We found that THC showed the ability to inhibit anarchist resistance in cholangiocarcinoma cells. Cell viability of cholangiocarcinoma treated with 20 micromolar THC significantly decreased at 12 hours after seeding. In addition, cell viability of cholangiocarcinoma treated with 40 micromolar THC was significantly decreased at 6 hours and 12 hours after seeding. We also evaluated the signaling pathway relevant to THC inhibition of cholangiocarcinoma cell progression. Our previous study showed that stimulation of CXCL12, CXCR4 plays an important role in cholangiocarcinoma cells invasion by induction of MEC1 and MEC2 and AKT signaling transduction and stimulation of actin polymerization. In this study, we suggested that the mechanism by which low-dose THC inhibit cholangiocarcinoma cells invasion involved in PIT AKT signaling pathway and MAP kinase signaling pathway by decreasing of the phosphorylation of MEC1 and MEC2 and phosphorylation of AKT. The next study is the effects of CBN on human lung cancer cells. CBN is a non-enzymatic oxidative byproduct of THC, mostly found in aged cannabis samples. CBN is a mildly psychoactive cannabinoid. CBN demonstrated anticonvulsant, anti-inflammatory, produce sedation combined with THC and inhibit some cancer cells. Firstly, we evaluated for the effects of CBN on lung cancer cell proliferation. Lung cancer cells were treated with CBN for 24 hours. This figure shows that lung cancer cells treated with CBN has significantly reduced the number of surviving cells after 24 hours compared to untreated cells. The results show that at high concentration of CBN from 40 to 100 micromolar, lung cancer cells were significantly inhibited in a dose-dependent manner. The effects of CBN on human lung cancer cell migration, we found that at 10 micromolar CBN, lung cancer cell migration significantly decreased compared to untreated cells at 12 to 48 hours. Apoptosis assays by flow cytometry using annexin 5 staining method was used to count the number of cells that have derived to apoptosis. Vehicle-treated lung cancer cells show a number of apoptotic cells, about 7% of total cells. CBN-treated cells at 10 micromolar and 20 micromolar show higher number of apoptotic cells compared to vehicle-treated cells. The study showed that at 20 micromolar CBN, number of apoptotic lung cancer cells were significantly increased. And then human lung cancer cells were used to induce tumor in nude mice by subcutaneous injection in a single dose. After cell injection for 2 to 3 weeks, human lung cancer cells develop a tumor at the site of injection. After tumors reach an appropriate size, mice were divided into 3 groups: vehicle-treated groups or control group, low-dose CBN-treated group, and high-dose CBN-treated group. Mice were given subcutaneous injection of CBN or vehicle for 3 weeks. This figure shows tumor size of vehicle-treated and CBN-treated mice. You can see that tumor of high-dose CBN-treated mouse was significantly reduced compared to vehicle-treated mouse and low-dose CBN-treated mouse. This figure shows the effects of CBN on tumor growth of xenograft mice. The blue line shows tumor growth of vehicle-treated mice. Green line shows tumor growth of low-dose CBN-treated mice. And red line shows tumor growth of high-dose CBN-treated mice. After a week, vehicle-treated group show rapid tumor growth compared to CBN-treated groups. After the second week, tumor growth of high-dose CBN-treated mice were significantly inhibited compared to that in the vehicle-treated mice. To confirm cancer cell characteristics, tumor from xenograft mice were determined for histopathology. Tumor sample revealed multifocal tubular adenocarcinoma, single or double layers of adenocarcinoma cells. In summary, cannabinoids demonstrated anti-tumor activity in vitro and in vivo. The molecular mechanisms of cannabinoids impair tumor progression by induction of cancer cell death, inhibition of angiogenesis, invasion, and metastasis. To the best of our knowledge, we suggested that cannabinoids may represent one potential approach to cancer therapy. Hope you enjoy staying at home. We will fight COVID-19 together. Thank you for watching.